Hello, I'm Collin with the U.S. Geological Survey. Thank you for choosing to view this presentation. I'll be talking about the Dragonfly Mercury Project, a national-scale citizen science program for monitoring mercury risk to ecosystem and human health. Before I begin, I would like to acknowledge the co-authors here on this title slide for all of their contributions, as well as the National Park Service and the park resource specialists that facilitated community engagement, and then finally the citizen scientists themselves that assisted across the country in this. The Dragonfly Mercury Project is a national scale study that spans landscapes with the goal of linking scientific efforts to understand the drivers and risks of mercury pollution with public engagement, collectively to inform management solutions or to inform on risk of mercury to environmental and human health. The dragonflies themselves are used as biological mercury indicators for several reasons. The dragonfly larvae is an aquatic form of the dragonfly prior to emerging as a winged adult. And they have extremely high site fidelity, representing very fine scale spatial extent of mercury exposure in aquatic food webs. They're also common and widespread, occurring in most freshwater habitats and across all 6 continents. They can inform on mercury condition in fishless waters, and they represent an important link in the food web, both as a voracious predator integrating the availability of mercury at lower trophic levels, and they represent a common fish prey item, providing mercury to predatory fish that eat them. And then they also serve as a vector to terrestrial habitats upon emerging as winged adults. They can transmit that methylmercury to wildlife in terrestrial environments where they may be consumed. Unlike many invertebrates, dragonflies do not have low or variable percent methylmercury, but most of the mercury in dragonfly tissues is in the methylmercury form, about 80%, and the variability is relatively low. Additionally, their large body size enables relatively easy identification. The sampling is relatively easy, requiring minimal gear or expertise. And their large size enables them to be analyzed as whole individuals so that one can gain an assessment of the variability in mercury concentrations in a population. Now, the Dragonfly Mercury Project is founded on a scientific basis, specifically to increase the understanding of mercury contamination as drivers, using dragonfly larvae as biosentinels of mercury bioaccumulation in ecosystems, but also to engage citizen scientists in the collection of dragonfly larvae and enhance science literacy. And collectively, these components serve to inform resource management decisions and provide guidance on communicating potential risk of mercury to humans and wildlife. Now, the program itself is designed to provide the Park Service resource management staff with all that they need to engage communities in sampling and providing the samples to us for analysis and interpretation later. And the success of this effort, which I'll talk a little bit about later, is really due to public participation, and nearly 2/3 of the Dragonfly Mercury Project citizen scientists are youth, largely high school groups and middle school groups, but also other youth programs, such as the Girl Scouts of America here in Glen Canyon National Recreation Area, or Urban Groundworks here in Shenandoah National Park. So we've engaged a broad coalition of citizen scientists across the country to really facilitate youth to both get educated and facilitate sampling and understanding the distribution of mercury in dragonflies at a scale that really wouldn't be feasible in any. Now, in terms of the history, the project started in 2009, relatively meager start with 3 parks and about 40 citizen scientists. It's since grown very dramatically, both in collaborators and number of parks and citizen scientists engaged. And here in 2020, we've now sampled more than 110 parks and engaged more than 5,000 citizen scientists across the country. And each year we embark in sampling probably 40 to 70 parks in any given year. So we have a pretty broad footprint of annual annual sampling that gives us a great snapshot of mercury condition in these natural laboratories across the country. Now, the process and information flow, the establishment of this program, we have 6 major steps here that include the initial step of park engagement, identifying locations and habitats for sampling, either based on that both inform the park needs in terms of resource management, but also provide information for broader scale analyses and assessments at a regional or national scale. The logistics and the design feed into both of those components. And then the Park Service resource specialists engage with the public to conduct the dragonfly sampling. The samples are then provided to the laboratory for chemical analysis, data management, data interpretation, and the scientific outreach and communication products, including making all of our data available online for these groups to go back and look at after they've done their analysis, and for anybody that's interested to download and examine the data for any questions that they may have about mercury cycling in the environment. And with that, I'll talk a little bit about the findings of our initial foundational paper that really established this program and illustrate the scope of the information that we are collecting. And this paper here, this is the paper here, recently published in Environmental Science and Technology. Now, with any bio, a really important component is that it's reflective of or an indicator of something that we care about. And here we sampled dragonfly larvae. Each dot represents a site paired with mercury concentrations in 4 different guilds of fish: piscivores, sunfish, salmonids, and forage fish, as well as 2 different taxa of amphibians, both salamanders and frogs. And we see that dragonfly mercury concentrations are correlated with all 4 guilds of fish, as well as the two types of amphibians. So these dragonfly mercury concentrations provide an index of the amount of mercury in different components of the food web and provide an integrated assessment of the exposure that an ecological community or humans that might be eating fish might be subjected to in these environments. Additionally, because we can predict mercury and fish concentration using dragonflies, we can estimate the potential risk to fish or birds that might eat those fish, or humans that might eat those fish based on established benchmarks. And we've done this here through an integrated impairment index. And you can see we have 5 levels of impairment ranging from sub-impairment to severe impairment. And we've essentially plotted the concentrations of mercury in fish that are associated with impairment at low impairment thresholds, moderate thresholds, and high thresholds for dietary exposure in fish, in situ tissue concentrations in fish, as well as dietary exposure to birds, and EPA methylmercury criterion for humans. And then identify the dragonfly concentrations associated with those benchmarks. And by doing so, we can look at the accumulation of exceedances in both number and severity. And through that, we've developed an impairment index that lets us look at the distribution of potential risk in the landscape. And here we've identified across more than 500 site years, or it might be 700 site years, the relative distribution of potential impairment to ecological communities, ranging from sites below known impairment benchmarks to those with severe ecological risk. And an important aspect of this is the variability with which you see the distribution of risk across the U.S. And in some parks, there are sites with both severe risk and that are below known benchmarks. So this highlights the variation that we know exists in mercury concentrations in the landscape. And with this, about 10 or 11% of the sites exceed high or severe impairment benchmarks. And similarly, about 10 or 11% are below any known impairment benchmark. But more than 50% exceed this moderate impairment benchmark. And that's an important one because that indicates that that top predator fish in these water bodies would exceed the EPA criterion for protection of human health. and so this indicates, a broad variability in potential risk across the U.S., as well as where those risk levels may be. So I hinted at the variability, spatial variability before, but you can really take that in with this distribution of data here. These are the geometric mean concentrations for each of the national park units. Here, the bar represents the park, and each dot represents a site within a park. And so across the entire U.S., we actually have a 135-fold range in Dragonfly mercury concentrations from the lowest site to the highest site. Now, this in contrast to, say, atmospheric deposition, where we have maybe a 10-fold range at most in sites with the highest and lowest mercury deposition. So that we know that landscapes play an important role in the production of methylmercury and bioaccumulation through the food web. This presents an example of that. And just further evidence of that is in these parks, such as Olympic National Park, Yellowstone, Mount Rainier, where we have extensive sampling. You can see that there is tremendous variation from one site to another, exceeding 30 to 40-fold variation from the lowest site to the highest site. And these sites are relatively close to each other. So this suggests that it's these in situ processes, the ecosystem level biogeochemical processes and food web processes that drive the variation across the landscape and that influence risk. And that's important because it indicates that although reductions in mercury loading are valuable, they're going to respond differently based on the habitat characteristics in the environment, and understanding the mechanisms by which we can control mercury methylation are another way of this. And this is also illustrated here in looking at mercury concentrations among habitats. So you can see that major habitat categories, lakes and ponds, had lower mercury than rivers and streams. And when we break that down even further, that those differences are even more pronounced when we look at sites here with, that have expansive wetland coverage in comparison to those with no surrounding wetlands. And you can see that those sites with expansive wetland coverage tend to have more mercury concentration, higher mercury concentrations than others. Similarly, within wetlands themselves, seasonal wetlands and tend to have higher concentrations than those that are permanently submerged, and that's consistent with evidence that we've seen in other studies elsewhere. So in conclusion, the 5 core elements for engagement and relevance to this project include risk quantification to help inform management solutions. Also, connection with effectiveness evaluation as we move forward with the Minamata Convention and reductions in global mercury emissions, testing how those reductions in emissions are translating to exposure in food webs, and then tying that to both education and outreach and communication, provide a really holistic approach to understanding the cycling of mercury and the potential risks on the landscape, while also engaging and educating our citizenry and our communities. So moving forward with this effort, we're beginning to build predictive models to identify and quantify the drivers to inform management. And that includes expanding habitat replication across the landscape and site replication within habitats, enhancing our understanding of ancillary parameters that influence mercury cycling and bioaccumulation, including geospatial layers, and then continuing our time series at select locations to look at temporal patterns of mercury bioaccumulation across the landscape. And with that, I look forward to any questions that you might have. Similarly, if you're interested in more information on this project, you can read the paper cited in there, or go to the project website, or we have an interactive story map that you can look at. And I would like to acknowledge the support of numerous partners in both resources and in-kind support to help get this work done. Thank you for your